Welcome to AP Biology. This video is an introduction to the cell membrane. The cell membrane, or the plasma membrane, is the boundary in between the inside of the cell and the outside of the cell, its external surroundings. The plasma membrane is selectively permeable, which means that it exerts some control over what can go through it. The cell membrane is a fluid mosaic of lipids and proteins. The most abundant lipid making up the cell membrane is the phospholipid, and the phospholipid is an amphipathic molecule. To be amphipathic means that you have a polar or hydrophilic region and a nonpolar or hydrophobic region. If you look at the phospholipid structure, it's made up of two fatty acids. These fatty acids are nonpolar or hydrophobic, and it has a polar or hydrophilic head. And the important part of that polar or hydrophilic head is this phosphate group here. When you put phospholipids in water, and 70% of our body is made up of water, they immediately arrange themselves into a bilayer. This is because like is attracted to like. The nonpolar hydrophobic tails are attracted to each other, so you have nonpolar next to nonpolar, and the polar heads are going to be attracted to the water on the inside and outside of the cell. So when you look at the cell membrane, or the plasma membrane, it's made up of a phospholipid bilayer, two layers of phospholipids, where the heads are facing outside the cell and inside the cell, and the tails are facing each other. The current model of the cell membrane is called the fluid mosaic model. When we use the term fluid, we're referring to these phospholipids. The mosaic aspect of the model of the cell membrane refers to the proteins that are embedded in the phospholipid bilayer. Here you see a picture of these proteins that span the entire cell membrane. How do we know that this is the structure of the cell membrane? We can use a technique called freeze fracture in order to study the structure of the cell membrane. Here we see a picture of the cell of freeze fracturing, and they basically freeze the cell membrane and they break it into two parts. They split apart the two layers of the cell membrane, and when you split it apart, you can see that one layer here, which would be the bottom, is going to have all these proteins sticking out of it, and the other side, which you see right here, is going to have all these holes. So you can see immediately that the cell membrane is made up of two layers of phospholipids, and that these proteins are going to span the entire membrane. Hence the fluid mosaic model. Here you see a picture of what the entire plasma membrane looks like. A lot of this we've studied in the past. Here you see our phospholipid bilayer, where the polar heads are facing out and facing in, and the nonpolar tails are facing each other. You also see the proteins that span the entire cell membrane. On the inside of the cell membrane, here we see the cortex, and the cortex are made up of those actin microfilaments that give a cell its characteristic shape. And on the outside of the cell here, we see the extracellular matrix, which is made up of different glycoproteins, the most important one that you see right here being collagen. You could also see that embedded within the cell membrane is going to be cholesterol, and we're going to see what the function of cholesterol is in a minute. The cell membrane is fluid, meaning that nothing is really locked into place. These phospholipids are constantly moving, and they constantly are drifting about laterally, meaning that they're not locked in place. They kind of can move back and forth, left and right. they very rarely flip-flop from the outside of the cell membrane to the inside of the cell membrane, but it does happen occasionally. It says here about once per month. So I kind of think of these phospholipids as being like buoys that are not tied down to the bottom of the ocean. They're just drifting around laterally. The proteins are also not locked into place. They're constantly drifting around as well. Here you see an experiment that shows this to be true. What they did was they fused two cells together, a human cell and a mouse cell, and you can see that the proteins are labeled with different colors. If the proteins were locked into place, when these two cells were fused together, you'd see all the pink on one side, you'd see all the gray on the other side. But after the two cells have been fused, you can see that the two proteins are equally distributed across both cells. Therefore, we know that they have to be mobile within that cell membrane. So you can see here what we've gone over really demonstrates the fluid mosaic model of the cell membrane. The fluidity referring to the fact that the phospholipids and the proteins are not locked into place, they're constantly drifting around. The mosaic refers to the patchwork of proteins that are embedded within the phospholipid bilayer. Now, it's important for the membrane to maintain the proper fluidity. Under cold conditions, the cell membrane would tend to become more solid because colder weather or colder temperatures restrict movement of the phospholipids. Under high temperatures, the cell membrane would tend to become too fluid. There'd be too much movement. Now, if you look at the cell membrane, in order to maintain the proper fluidity, the cell membrane can alter the amount of saturated fatty acids versus unsaturated fatty acids in the phospholipids. You can see on the left here that a cell membrane that is more rich in unsaturated fatty acids is going to be more liquid at room temperature. You can see that phospholipids that contain all saturated fatty acids are going to be more solid at room temperature. Well, how does this change with different types of organisms? Organisms that live in colder environments where the cell membranes would tend to become too solid, they're going to have cell membranes more rich in unsaturated fatty acids. Organisms that live in hot environments where the cell membrane would tend to become too fluid, there'd be too much movement, are going to have phospholipids that are more rich in saturated fatty acids to prevent it from becoming too liquid. Besides having a certain ratio of saturated versus unsaturated fatty acids in order to maintain the proper fluidity, cholesterol is also going to play a role in maintaining the proper fluidity. Under cold temperatures, the cholesterol embedded in the cell membrane is going to prevent the phospholipids from packing too closely together. And under high temperatures, the cholesterol is going to restrict movement of the phospholipids. So cholesterol is also a really important molecule embedded within the cell membrane that is used to help maintain the proper fluidity. There are many different types of proteins that are associated with the cell membrane. There are two major types, one being the integral protein and another being a peripheral protein. Peripheral proteins do not span the entire membrane. They're either found on the outside of the membrane or they're found on the inside of the membrane. Integral proteins span the entire plasma membrane. So if you look at the structure of an integral protein, it needs to have hydrophilic regions and it needs to have hydrophobic regions. The regions exposed to the inside and outside of the cell have got to be hydrophilic, And this core middle part here that's exposed to the phospholipid tails, the fatty acid tails, needs to be hydrophobic. There are 6 major functions of these membrane proteins. 1, these membrane proteins are going to be involved with transport. They're going to be used to move large or polar things into and out of the cell. 2, these proteins can function as enzymes. They can catalyze chemical reactions happening at the cell membrane. 3, they can function as receptors. They can be involved with signal transduction pathways, allowing one cell to communicate with another cell. 4, they're going to be involved with cell-to-cell recognition. It's going to be how our white blood cells, members of the immune system, are able to tell what cells belong in our body and what cells do not belong in our body. 5, they can be involved with intercellular joining. These proteins can be used to attach cells together. We saw them before with desmosomes or tight junctions. Those are all made using membrane proteins. And finally, 6, we see attachment of the cytoskeleton to the extracellular matrix. Right here you see that the actin microfilaments inside the cell are attached to the collagen and other glycoproteins outside the cell using these membrane proteins that you see right here. All of the important functions, all the important things happening at the cell membrane are going to take place using these membrane proteins. They are so important to understand. Many times carbohydrates are associated with the proteins on the cell membrane. When proteins have a carbohydrate attached to it, we call this structure a glycoprotein. These glycoproteins are particularly important with the process of cell-to-cell recognition. It's how the white blood cells in our body are able to distinguish what cells belong inside of our body versus foreign cells like foreign bacteria that can be infecting our body. Here you see a picture of the glycoprotein that we saw before. Here's the protein, here's the carbohydrate attached to it. And these carbohydrates are like billboards on the outside of our cell. They are a sign that distinguishes that cell as belonging to you. Here you see the cell membrane of the white blood cell, and it's able to use a protein on its cell membrane in order to read that sign and be able to distinguish whether or not that cell belongs inside of us. Here you see an example of how the receptors on the cell membrane are so important. Receptors are another example of a protein attached to the cell membrane. And it's how one cell in the body is going to be able to communicate with another cell. Cells often release chemicals, we call them ligands, which are going to be released from the cell and bind to the receptor on another cell, and it's going to transmit a message. Here you see an example of some research that's being currently done using these receptors. The HIV virus, when it infects its host white blood cell, it gains entry into the white blood cell by first docking with a pair of receptors on the cell membrane. They're called the CD4 receptor and the CCR5. Scientists have discovered that some people naturally do not contain the CCR5 receptor. Without this pair of receptors, the HIV virus cannot gain entry, cannot infect its host white blood cell. So what scientists are doing is they're working on a competitive inhibitor, an inhibitor that is going to bind to the CCR5 protein and block it. If you block the CCR5 protein, effectively the cell does not contain it, and HIV will not be able to infect that cell. This concludes this video.